Thank you very much for coming, and thank you all the people that are online. Some of you know about me, some of you maybe don't know a lot about me. So I'm Vivian. I'm an entomologist, and I've been very uh, like interested in fungi because I've been studying insects that are eating mushrooms. So that started in 2009 when I was doing my PhD in Brazil. Um, Previously, I was working with ants. I've been working with, can they hear me? I don't know. I've been working with ants. Can you hear me? Okay. Yes, it's because I can't hear me because of this. So it's a little bit weird. Yes. Sorry. Okay. That's better, I think. Yes. Okay. Yes, that's better. That's better. Can hear me better. So yes, I was working previously with ants that cultivate fungi, and then I moved to beetles that were eating usually polypores. So that's why I'm very interested. So I started collecting polypores in 2009 2000, yes, not a long time ago. Now I'm here in Australia. At this moment, I'm not working with fungi and my beetles. I'm doing that as a like just my own research, a volunteer staff at Queensland Museum. They are helping me a lot. But yes, I'm still like very happy of being involved with my like second research projects, and during my full time, like I have a job working with fruit flies. So yes, it's quite different. I'm killing fruit flies, but I'm still very interested in continue all my research that I studied in Brazil. And now I have a lot of material from Australia, thanks to many of QMS members that have been helping me collecting mushrooms, collecting polypores, and also all the forests that I've been also going with most of you. So today I want to share a little bit of what we can know, what you don't know. I think that most of you don't know what's inside these mushrooms or this what is a fungi. So it's a completely unseen world because most of these the little organisms are too little that you can notice, and some of them like people that are mycologists, some of them consider them like as a pe- pest of mushrooms, but they have a very important um, role in the environment. So I will talk a little bit more about that too. Um, Most of the photos here were taken by me. When they are not taken by me, I'm putting the name of the person. I have lots from Wayne. I put it Wayne's name. Well, thank you very much. I will start with this nice um, phrase. So this is an artwork that I asked to a friend to make for this occasion. Like I gave this talk like a month ago for the Queensland Naturalist Club, and yes, that was made just to work with my talk about what I'm doing. But yes, this is quite nice. Like, why did the mushroom go to the party? Like, you know, all of you know this phrase because he is a fungi. So that's just to try to start with something different. So this is an artwork. The artwork was made by Abby McClure. She's an Australian artist, and she has a small business, and she sells some nice cars and other stuff. So yes, I'm always trying to support the small businesses. So yes, she made this just for this occasion. So what I'm going to talk is first, what are fungi? So this is really weird, would be really weird for me because most of you know more than me about fungi. So it would be a very quickly talk about what's fungi. Then I will try to go deeper in what I know more, that is what is mycophagy. So what are the organisms that are eating fungi? What are the interactions that we know at this moment? what are who are the mycetobionts? So that's a term that some of you might know, some of you won't. What's important of this interaction? The techniques for collecting. So you know, all most of you that have been in QMS for a long time and have come to our workshops know how to collect mushrooms. But I will show you how to collect the little insects that are inside them. So that's a little bit different. And at the end, I will talk about my research. Um, okay. So well, I just brought this example. So So mushrooms, the fungi are the largest. So we have one that is considered the largest, are the oldest organism on the earth. Yes, so that's not a um, elephant or a blue whale. Yes, it's a fungi. It's a armillaria. So this is just the fruiting body. Of course, I put a little person underneath. It doesn't mean that the whole fruiting body is this high. No, it's just to show that is the all the mycelium that is all spread around the area that is in the United States. So it's covering an area that is more than 10 square kilometers. That's huge. Yes, it's considered that it could be have an age of 8,650 years. So it's very old, the oldest, the largest, and it's a fungi. So yes, a lot of people think that yeah, they're just here in this piece of wood. No, they can be spread all around. They have we have all this connection under the ground, and that will give us a lot of things to talk about because they can be doing lots of things in different areas. Yes, of course, the importance is decomposition, and we need them to generate soil and new nutrients also to the rest of the organism. So what are fungi? Well, I can say a million of things of what are fungi, but this is a nice photo that we took in one of the last forests. I think that was Bennett. Can't remember. Yeah, so you can see here. So in this photo we can see mycelium in the bottom. We can see the substrate that were um, um, leaf litter, but also we have the fruiting bodies. So fungi are pluricellular organisms. They are not plants. We thought a long time ago that they were related to plants, but they are not. They are heterotrophs. They need other, they eat other stuff. They don't produce their own food, of course. They have chitin in their wall cells. They produce these very nice structures that will be the vegetative growth structures that we call as hyphae. That's unique in within mushroom, yeah, within this big group that I will show you later. And they will all together, they will form what we call mycelium. And what hyphae are doing are just going through the substrate, whatever kind of substrate they are using, and they will be absorbing yes, nutrients and taking also nutrients, absorbing water and taking nutrients between all this like a web that they are making. Well, I could say a lot of things about what else are fungi. They produce spores in like will be the reproductive phase. Some of them have these nice fruiting bodies, and some of them they will change. The shapes will change. The way that it is distributed usually is like by wind, by water. So they don't move. What moves like what is dispersed are like the spores, but also the vegetative part. The hyphae will be growing, and that's why they can like disperse also. So well, I can't remember many things about that. So I will go to the next one. So we have this is a tree of life showing where fungi are, and we can see. I will show here for people that fungi are very related, are more related to animals to the rest of what we thought they were related, like plants, for example. So we are like the closest relative to fungi, and we have many things in common too. We have this slime mold will be like a separate group. That's what a lot of people say that now they are not fungi. Um, yes, sorry. Yes, sorry. It's hard to explain here and here. Yes, but you can see that. So we have a little bit, I brought a photo of slime molds. We took that photo also in one of our forests. So these are not true fungi. And we have, so there's a group called Eumycota that would be the group of true fungi. But we have separate groups. So this is also a phylogenetic tree based on DNA, on RNA genes showing the 5 true phyla in the group of fungi. So we have water moles that will be Crytridiomycota, it's hard to say. We have Zygomycota that will be the bread moles, Glycoromycota, and we have these, what we will know and we can find usually in our forests, and yes, what we usually call the fungi, that we will have the group of Basidiomycota and Ascomycota. So will be polypores, Basidiomycota will be all the ones that we can usually recognize as stinkhorns, polypore, mushrooms, and as Comicota we have yeast and morel, Shilarias, well, other groups. So that's like a abstract of what is that big tree. So we have here, I brought some photos like of what we call Basimyrmicota, and what I will be talking more is about this, because usually, so Basidiomycota, yes, than Ascomycota, because yes, they have these big fruiting bodies, and usually the insects, well, the group of insects that I've been working, they are like specialized in eating them. So the fruiting bodies, I brought here the fruiting bodies. So what's we call basidiom also, they have a pileus, that will be the cap. This is a very, very important structure because in the underside, usually we have the hymenophore, that is the tissue that bears the spores. We have also some of them, we have a nice stipe, some of them won't, or stem. And depending also of the shape and the quality of like how hard it is, will be the group of insects or other invertebrates that are eating these fruiting bodies. So I brought 2 examples of some perennial fungi. So we have some polypores. So they will be growing slowly every year. So and we have also annual fungi that will be like just releasing, growing new fruiting bodies once a year, like usually when it's very wet, and then they will just disappear. They will don't die because, well, just disappear. But well, of course, the rest, the vegetative part will be under the soil. But yes, when it's time to spread and time to reproduce, they will produce these fruiting bodies. So, and these particular cases, when they are perennial and they are very hard, so we will be special group of insects eating these guys. And when they are fleshy and very, very like flimsy and watery, they will have other group of insects also. Also, when they last for a long time, so they will usually have organisms that have a life cycle, like a very, very long life cycle. They will develop slowly compared with the ones that are in annual fungi that maybe like one of these mushrooms will last just some days, so they have developed different characteristics to grow very, very fast compared with other groups. So talking about a little bit about eating fungi. So, well, this is like this is eating fungi, but for people. I just brought um, this slide because yes, so fungi are delicious. Yes, but yes, is we can and we are talking this a lot in our meetings and also in when we receive requests of identification and people say, "Can I eat them?" Well, some of them you can, but it's better not to do it. Yes, they won't. We have lots that are toxic, that we have lots that are very similar with non-toxic, but if you are not an expert and you don't have all these skills and you don't know even if this how is the soil, if they are like accumulating some toxic stuff, it's better not to do it. So I just brought that. But we have lots of organisms that are eating even the toxic, the ones that are toxic for vertebrates, you know, the ones that are toxic for humans or other animals. So yes, so I just brought that because as I told you, I gave this talk to a group of non-mycologists. So this good because a lot of people are always asking, can I eat? Can I eat? So this is an example of some of the common mushroom that we can find around Brisbane, in Brisbane Parks, that is green gill lepiota. So this can be usual. Like a lot of people, they think that they're similar with mushrooms. They're similar with some of the we can find in the supermarket. Maybe even when they are little ones, that they are quite similar. So yeah, that's they can be. But yes, underneath you can see here. It's very little this photo, but yeah, they have the red, the green spores. So that's one way to recognize it, and these guys are not edible. And I brought this photo because I thought that it was quite different. It was in my backyard and they were all together. So some friends said that it seemed like a, I don't know, weird fungi. So what's mycophagy? So mycophagy, of course, organisms that eat fungi. Um, they could be eating different parts of the fungi. So they could be eating fruiting bodies, the spores, or mycelium. Some can eat everything. Some of them will eat just. Some of them can be specialized eating just mycelium, like just sucking liquids from hyphae. Some of them are yes. I will show you later. Some of them just eat spores, and they are very happy in the gills underneath the some mushrooms, eating just spore very fast. So yes, yes it depends of the organism that we are talking about. I just brought this example. It's a polypore, it's a ganoderma, and these little black dots that you can see in the photo and you can see here. They are tiny beetles that have around 2 millimeters, 2.5 millimeters. So this is one of the groups that I've been studying since I started working with insect fungi interaction. Um, so here I have an example of the ones that are eating spores. So this is a tiny little roe beetle that you can see on the top. Um, it's called Garofina. It's common in oyster mushrooms. So they have a special mouth parts to be just eating. The position of the mouth part will make it just eating perfectly around all these like highways between the gills of the oyster mushrooms. And these guys are also so amazing that some species will lay instead of lay egg, will lay larvae, because like yeah, many of the oyster mushrooms won't last a lot, so they need a very fast growing, so they won't lay eggs. They will lay already a baby baby larvae, and they will be ready to eat what they have at this moment. um, we have guys that are eating just mycelium. So that's a flat back, is a family Aradidae. So they have a very long stylus that is like a tube, and they will be sucking, so they can go inside the wood find where the mycelium is, where they get, and put that is still two things that will go through a hyphae, and they will just suck in like drinking, I don't know, soda, drinking water, sucking all the contents. Like that's fine because as the water is running uh, within the inside the hyphae, so they will just sit and all these fluids will come in, and they can stay there hours, days. Like it's amazing. Some of the cell is so big that like they are very vulnerable to predators, but that's why they seem they are like a piece of wood. They are very hard to see because they sit on top of the piece of like rotten wood, doing that for hours and hours and hours, and as it's too long, they won't be able to remove it easily. So yes, but well, they can do it. So what they are sucking water from straight from the mycelium. They will be peeing all the time. Yes, they are. So what are the interactions that we can have? So every organism that have a kind of interaction with a mushroom or fungi, we call mycetocols. Um, beg your pardon if I don't pronounce well. I try to, sometimes I'm trying to remember the weight, but yes, it's quite hard for me. I've been speaking English just for 3 years. So mycetocols, they will have some kind of relation. It doesn't mean what kind. But all the mycetocols we can divide in three big groups. So mycetoxins are the ones that have a little bit of interaction. So if maybe like this guy just sit on top and wait for another insect to come and will eat it. You know, but they need to be sitting because the other, like their prey, will be on top of mushrooms. So that's an example. My cetophiles are the ones that they really do like to be there. So they will be there just during some season or while they are larvae or young, and adults they will go and do another thing. And my mycetobionts are the ones that will be living inside fungi or up and well down well, everywhere. Like, they are very related with fungi the whole life cycle, the whole life. So from egg to adult, and they will disperse to find more fungi. Yes, so not they won't leave that. So that's their only way of living. Um, so I have here example. Um, so we have a porcelain mushroom. So that's the Manciella that we collected, and I tried to identify. Like we said that it was another species. I don't know if you remember that. That was for the when with my mom. That we get all these ticks. Well, I got a lot of ticks. So that was in a very nice wood, like in making like a bridge. We took this photo. So you can see lots of little flies on top. So these flies, they usually will be like mycetobionts because larvae from adults will be eating what is in there, eating the fruiting body. But I put there's another big fly here. You see another big fly here. So that fly doesn't like, it's not related with that. I'm not sure what she was doing. Yes, but this is a banana stalk fly. So usually they will be eating rotten trunks. Like they lay eggs, the larvae will grow not in the banana stalk. You know, so well maybe looking around or just drinking water. Yes, but it's have some kind of relation. But you will see. So then we took a photo underneath, and this is what I found. So we have an ant, a green head ant. So this ant is a predator, and she was just trying to eat some little flies, or maybe some fly larvae that were inside. And also we have here a nice looking beetle. You can see that beetle? That is a pleasing fungus beetle. So These guys are mycetobionts. So it's a family Erotilidae. They are very, very nice, very beautiful beetles. And all the larvae will be eating the flesh of this mushroom, but also they can eat the spores. Larvae and adults will be completely related to this fungi. So depending of the kind of fungi, as I told you, will be the kind of insects that will be using that kind of food. So we have usually agaricot fungi and boletes, the fleshy ones, will be more colonized by flies. And the ones that are hard as a rock, like polypores, that we call dry and dense allotrophic fungi, they will be colonized by beetles. And they will have all these mouth parts, very, very specialized to this kind of food that they are eating. Um, this is another example of relation. So we have lots of flies, you know that flies are attracted to stinghorns. So stinghorns, they release the spores in kind of like a jelly stuff that we call gleba. So the spores are here, but they are like in a liquid thing, and that it's producing a very strong, or how do you call stingy? Yes, that's why they put this thing. That's the odor will attract flies. The flies, yes, we have lots of different group. Like here in this photo. This photo was done, passed by Jeff Montis to me. So we have 1, 2, 3, 4 different families of flies that were attracted to this stinkhorn. And you can see in the photo, like this is day 1 and the other is day 2. So they come quickly. So when it grows, it emerges, and they will lick all this gleba, and then the cap will be just completely clear. Do you know what's the reason of this skirt? Because I don't know if you've thought about, wow, this is so beautiful. It is so beautiful. But the scare, the paper of the role of this scare is to protect these flies from being predated by other insects. So the ants usually will be the predators, like normal predators. So an ant will crawl, for example, through this okay, stalk, and it will go down and then up and down, and never go up because it has this scare. So they are not so smart to say, oh no, if I try to go up to the scare, no, they won't do it. So that will protect the flies. So that's a mechanism that the mushroom evolved. That's amazing. And if you find, and also Jeff in that little article that he wrote about this, um, he found some stinghorns that had problems. They didn't develop a very good skirt, and lots of ants were on top eating the flies. So that's another very nice. So they need the flies to disperse their spores. So they are also creating mechanisms to protect them. Talking a little bit about the beetles. So the beetles will be the ones eating more dark stuff, more hard stuff. So we have around 80 families. So in total, we have almost 400,000 species of beetles. So it's the most abundant and most diverse group of insects. Well, there are new research saying that maybe the parasitoids of the insects will be even more diverse. So, for example, um, tiny little wasps, people say that will be in the future. Well, we need more research and more people doing taxonomy of them, and even a lot of people say that flies can be also. But well, at this moment, beetles are still the most diverse group of insects, and we have 80 families that are they have a strong relation with um, fungi and plant material that was altered by these enzymes of the fungi, but just 25 families that are strictly mycophagous. That so the other families will be eating a little bit or like using like having a strong interaction, but they are not just eating fungi. They can eat other stuff. So 25 families are mycophagous in the strict sense. Um, so I have some examples. So for example, this guy, it's a beautiful tenebrionid. His name is Birsas macleji. It has these very beautiful horns. That's a male. So they are eating polypus. They are usually a little bit gregarious. Males have this huge horn, and they will also produce a very strong smell to protect themselves from predation. Um, yes, this can be found. Like it's very easy to find here in the polypore samples that I've been collecting around. Is very common. Yes, they are quite small. Will be around I don't know 3 centimeters, so not too big. Centimeters. Yeah, no one. No, like 2 centimeters. No, millimeter were too tiny. Like the yes. No, these are more like my insects usually, my beetles that you will see. I don't have this guy here. Um, so yes, my beetles are usually this other family are 2 millimeters. So this is 2 millimeters. One guy like this would be like this, that is like 1 to 2 centimeters. Yes, that's quite bigger compared. So the mycetobionts insects will be the ones that will be, as I told you, they will be using fungi as habitat, food, place for oviposition, and also shelter. Um, so the larvae, I have an example. So this guy was the first photo that I show you eating this nice Ganoderma. The larvae will be inside, but the adults also will be inside. They will be making chambers for their larvae, but also will be protecting their territory. We have different associations that we've been finding here in Australia. So for example, on top is an hexagonia, and you can see a hexagonia with this fluffy stuff hanging from. So this photo, Colleen Foils, it's a lady from Queensland Museum, sent it to me, and I remember that I sent to some members because no one knew at that moment. Sorry, what was this fluffy stuff? If this was like it was something related with the growing of the fungi, or if it was insect stuff. And yes, I've been collecting these guys and studying. So this is the um, frass of a little beetle, like it will be the larvae, produce a frass in like threads, and they use it to protect themselves. So they are making this fluffy stuff, and they live inside. They will be eating the pores. They will be underneath the polypores. So they were eating all the what can they eat from the pores. Yes, eating spores and a little bit of flesh also. So that's a very nice interaction, and you might find this like if you like have more attention when you're collecting. That's very interesting. Like I've been trying to rear these guys to adults. I have some ones that they did very well, but I haven't been able to record or like to know how many life stages have in during the larval cycle or things like that. So yes, it needs to be. I will need like I need more samples. So I had just 3 different samples, so that's nothing for a taxonomic study. So yes, no one has studied that in Australia before. We have also the other example of the little case moths that Wayne photographed with was with Judith. Was Yes, last year, the end of last year. So it was another polypore, this time a Ganoderma, with these little tubes hanging from the underneath, and they were moving. They were moving a lot. So what? So I got these samples from Wayne. So it still is a mystery case bearing caterpillar. So I sent. So it's a caterpillar. So I remove it from the little case. Is this what you can see here at the bottom? Um, it's a little bit strange from what is usually known from polypores. So we have several tinies that is a group of moths that are more common in polypores, but this guy is like in between psychid, that is the ones that produce this
I'm
nice
listening to a
case,
talk by
and the guys.
Dr. Vivian
Sorry.
Sandoval.
Okay. It's a lot of noise. Sorry, if you can mute, please. Thank you very much. Um, well, so we have these, I still have these caterpillars. That's also something unusual. That's something that, like, I think um, some entomologists have um, said to me that yes, never saw that before. Some of them said, "Oh yes, I've seen these tubes in just in wood, in like in rotten wood." So yes, they must be also eating. So they were here eating the polypore. They eating the underneath on the polypore. They make these spots here are the marks that the mouth parts leave. And so they eat, and then they move and make another hole, and then move, eat all what they can eat from the like surface, and then move. But Jeff Monteith said to me that he found the same guys in a piece of rotten wood, so maybe eating mycelium, because he didn't see any like fruiting body. So, but they must be very related, like of course related to fungi. We don't know what they are. We need some adults, like we need adults, because these some group of insects are very different, difficult to identify. More when they're tiny, because they are tiny, like also yes, little cases. So that's why it's so important for me and for many entomologists that mycologists that find something weird in their sample will let us know, because yes, some of things that yeah, you will never find this in an entomological collection, because they are living in the fungi without inside fruiting bodies, so yes, it's quite important. So what's left? So yes, so all these guys have this relation, they're eating, living inside, but at the end what we have, oh, I think that is a bit block. What's left over. So I will talk while it covers. So we have here, oh yes, here. So this is an example of some like this, is too big. So we have here a very old Ganoderma. I got all these thing from a lady that she requests, she sent us a message saying that one of the trees of her backyard was a little bit weird, and the arborist asked to cut it, and they have these polypores in the base. So I said to her, well, if you're going to cut it, can you please collect the polypores before the arborist just put it into the machine to make mulch. So she brought all this to me. And yes, it's full of tiny little holes in the back. It's similar with the photo that I'm showing in this slide. So we have lots of holes. And there are tiny insects, like beetles will be beetles doing these holes, 1 to 2 millimeters. They will be eating this. So at the end of many years, yes, because imagine the size of the insects and the size of the fruiting bodies, are some of them are huge. We will have just frass, what we can see. And I have here, if you can after we finish, you can see here, I have lots of frass, just dust. So all these polypore will be turned into dust. Yes, and that's very important the health of the environment. That's very important because so these guys are polypores or whatever kind of fungi. They are removing nutrients from dead wood or from leaves or whatever substrate they are using. And all these nutrients are being stored here also or transformed into other kind of nutrients. And then if we won't have insects doing this job, or insects or even other invertebrates, because we have, I'm just talking about the insect, but we have snails, lots of snails doing this job also, but in the most fleshy mushroom. We have springtails that are not insects. A lot of people think, but they are not insects. Well, we have many group of invertebrates that are doing this job. So if no one will be using this food, this will be just stored forever, like more with these very hardy fruiting bodies. So after some years or maybe some months, depending how many insects we have, we will have just like the pillars, like the outside, that will be the hardest part that they can eat, but the inside will be turned into dust that will be just frass, and this frass will come to the soil, and the soil will be more rich than before. um, and they did the same. They turn they turn a tree or a plant into something that will turn into material to make more soil. Yes, we can. Yeah, so they are very important for the functioning of ecosystems, nutrient recycling, as I told, and also food chains, because we have lots of other invertebrates eating these insects. So we have lots of spiders, myriapods, that will be the centipedes that also are predators, well, ants, well, other group of invertebrates doing that, but also birds and other vertebrates like little marsupials also will eat these insects. So yes, that's very important. Maybe, but they have also bad importance. Like they have an economical importance, and that is one of the reasons that can bring more attention to these guys, because they can be pests of commercial dried fungi. For example, these specific tiny little beetles that are eating polypores, but also they can be pests of commercial mushrooms, like oyster mushrooms. I have one, like when I arrived to Australia, like 2 years ago, had this message from this lady in Maleni that she is growing oyster mushroom, and she said that they have a big infestation of tiny little things walking around the oysters, and they lost the whole production that they have at that time. It was a lot, and they were these little beetles that I told you that they run and they eat and they lay larvae. Their life cycle is too fast, so they were just coming. They came into their shed where they have the oysters, and well, we're doing the job because they are usually outside eating mushrooms, other kind of mushrooms. So yes, well, what we can do, but yes, we can. So we need to study these guys also, because they can be very bad for mushrooms that have an economic importance. So also they are very bad for fungi collection. So in the herbariums, um, I didn't hear anything from here, so I asked when I went to the herbariums for a little while as a volunteer, and I didn't hear anything about that they had problems with these kind of beetles as pests. But I have many messages from all over the world, so people know that I'm working with these guys. Like 2 years ago, a lady from Mexico, she said that they were having a huge infestation in a huge herbarium in Mexico. They have the fungi collection, and they didn't know what to do. The problem is that if they spray and if they are inside the fruiting bodies, it will come in. It's the same that we have, the same problem that we have with pests in wood. Like it's very hard to get in, like the chemical to get into the wood, and these polypores are like good or dry, whatever dry fungi will be too dry to the chemical to get in. So yes, it need to be stored in a different way. They need to have different treatment. And when I was in Brazil doing my research with my students, we were at the beginning following the protocols that all of you do with mushrooms, like when you dry mushrooms, so around 40 degrees to put it into the, uh, dry the samples at 40 degrees, that's right? To In order to take them, dry it and then take it to the herbarium. But 40 degrees wasn't enough to kill these beetles. So yes, because I was always collecting, so I reserve like a voucher to put into the herbarium, and I was reserving one like at that temperature, and some weeks later it was just dust because they were still alive. So we need to freeze them first. Otherwise, it won't work. They will be inside eating. And I've seen a lot of people, even here in Brisbane, some mushroom growers, that they use as displays this beautiful Ganoderma or beautiful. I said, oh, it won't last a lot because they are inside. You can see the holes. If you see holes, there's someone inside. Must be a beetle or must be a caterpillar, like a moth larvae, will be doing its job. So yes, they need to be stored, like need to be freeze first. First, if we want to kill what is inside. Yeah. So sorry. Well, yeah. So for entomological purposes, like usually when we enter new material in a collection, we leave at least for a week in the freezer. If we have an ultra freezer, will be like a week. An ultra freezer will be good. If we don't have ultra freezer, people will leave it up to 3 weeks. So it depends on how big is your fridge or your the place where you're storing samples. So yes, another nice thing is that they can be invasive, and I put here that so Australia has been exported beetles to these tiny little beetles overseas. So we have this example. So it's Cisbilla melato. This is a tiny beetle. It's around 1.5 millimeters, eating usually eating Fomitopsis betulina in Europe. Yes, but it was here before. So in Europe is using that. I don't remember what I think that do we have Fomitopsis here? Yes, so might be using another species here because the problem is that we don't have a lot of records of the host records because we don't entomologists usually don't know how to identify fungi. Mycologists, they don't identify insects. So we have this. So when I have samples from the museum, they just said dry fungi or an bracket fungi, that's all. So that's very, very hard to do these links. But this, so in at the in 1900s, it seems that some samples that from the herbarium were sent from Australia to UK, and then well, these guys arrived alive. They were still alive. I told you they're really, really hard to kill, and they were just spread. They found something similar to eat that was this Fomitopsis betulina, and now, like in this map, I'm showing from 1900 to 2000. Now they're all spread around UK. I'm not sure if they went to the rest of Europe because no one else is studying. So that was a study by Brenda Ordlich, but she's not working with that anymore. So I'm not sure what's happening now with these guys. But yes, they were Australian. So there is a species in Australia. It's not so common here, but there is really common. And the techniques, and I forgot to bring my tools. But you've seen me, like a lot of people that have been in the forest have seen. So I usually put a little net underneath the bracket fungi, or even if it's a little mushroom or like bolete, whatever mushroom, I'd also try to put something underneath because they jump. So some groups won't because they are very, very inside, very deep, for example, in brackets. But the ones that are just eating the spores underneath in the lamellae, that they were just eating and walking, they will jump immediately. So I need to put something underneath. Will be a net or a little like a plastic container. And so this like study is very, very nice because It's like fungi are very abundant and easily to identify if we have like some mycologists, very good mycologists, people that know about it around. Also, the insects are easy to collect. I don't need, for example, to spend a lot of money with collection techniques or special traps. Now it's just by hand that we do that. They can be easily reared at home or at lab. I don't like, I'm doing my research at home. When I'm going to the museum, I just taking specimens to have a look under the microscope, but I just have them in small containers as I brought here also. They need to be very dry. This is for the polypores usually, well, the woolly ones. And if I'm starting from oysters and from fleshy mushrooms, it's better to put it in soil, like in a container with a little bit of soil. So that will keep it. Of course, they will get rotten in 2 days, but that will give me a little bit of time to rear them, to open and remove the insects that are inside. And sometimes I'm lucky to have adults, so there will be enough time for them to pupate, to jump to the soil and become adults. So yes, they won't take a lot of space also, even in a collection, because as I show you, they are tiny. You will have a look later. I have one here. So I put a beetle in a piece of paper and in a pin. You will have a look. But you can see, people that is online, you can see a entomological box here with lots of tiny pins. I don't know if they are seeing. Ah, it's in the other side. Okay, oh, that's so hard. Sorry. So yes, you can see here it's a collection of pins with a little bit of paper, and the black dots are insects that were reared from these different samples of fungi. And I'm trying to, like the ones that are from polypores, and remove the humidity with silica gel. So the same technique that we use when we collect samples for the herbarium. Um, yes, a little bit of so the research that I've been doing. Well, I talk a lot, but I have at this moment I'm working on 3 different projects. One with a genus called Xylographus, that they are all over the world. I studied them before in South America, Africa, and now I'm doing the Australian part. I have, it is not very diverse in Australia. I think that, yeah, there are just 4 or 5 new species, and one that is already described. So that's one of the projects, and there are lots of specimens from the museum. And also one of them, it was intercepted by biosecurity in the border. So it came from in commercial dried mushrooms from China. So that's the only thing that I know about it. Is this from China? It came in a dry mushroom. It's a new species. And a few months ago, I've got a message from a guy from China. He was requesting me to identify some samples just with photos of these little beetles. He was running a project over there, and he sent me the exact beetle that we found here in these Chinese samples. Is the same species. As he even sent me like very, very good quality photos. And so now I know that they are really from China. But the problem is that they did. He asked to his boss if I could use the locality to put it in my paper, because that will be like a very good um, way to describe a new species, saying that, look, they collected this, and already they send me the genitalia photo, because that's very important to go to species level. But they didn't. They said that no, that I'm not allowed to do it. So they don't want to um, give any names or any places in China for this sample. So I'm describing these species, but I will just say that they came from China. I can't say anything else. Um, and I have another specimen that was collected in China from another person. I found that in Brazil, but this person also didn't allow me to use it for my paper. So it's very hard sometimes because they are considered invasive species, so that could be very bad for China because of the problems with trading mushrooms to Australia and also pest beetles. So well, what can I do? Yes, so the other two projects, I have one project with um, this guy, Platydema, that are the little tenebrionids that make these threads. So I've been working with them, trying to study their life cycles. So this is the other project. And the other project is Gyrophina. They are the ones that are pests of oyster mushrooms. So there's no registers for them in Australia at this moment. So I've been collecting. So that's why I'm going to all these forests. Usually, I have a permit also. I have a permit of the Entomological Society, so that's why we have a permit for fungi, but also I have a permit to collect the little insects, and I'm collecting all sorts of raw beetles that can be eating bullets and other kind of agarics and yes, of fleshy mushrooms, because I want to see if the guys that were in the commercial oyster mushrooms were the same that are usually around the place or around Brisbane or around Queensland, or if it's just a particular species that is doing that, you know. So that's, yes, those are the three projects. I'm running very slow because, of course, I don't have a job working with that, so I'm doing that in my spare time. But I'm really, really happy with all like all new samples. A lot of people remembers me and send me photos. So yes, thank you. So I'm just requesting a live or dead bracket fungi or mushrooms, yes, with signs of insect infestation. So with holes or grabs, a lot of people have sent me things, and yes, I very, very appreciate that, because that is very important for me as like a entomologist working with um, mushroom samples and yes, polypore samples. So well, these are some photos. These are not from Australia. These are from Brazil when I was there. Thank you very much. I think that I took a lot. This is yes. So this is this little larva. I don't know what they are. Yes, I try to keep them. I try to identify, and the same to several samples to specialists, and they said, "Oh, you're the expert. You need to try to rear them and see what they are." So yes, it's quite hard sometimes because it's a whole universe. You don't know what they can be. Thank you. So I have a little back shop. It's like mycological and entomological and mycological shop selling jewelry. So I started making jewelry like when I arrived to Australia, and then I started making a lot of partnerships, and I'm usually like working with like at least 10 different artists in Australia. They are making things that I can't make. So so yeah, just want to say thank you, because I've got some orders. So usually, and I'm usually donating, like I'm supporting nonprofit organizations. So I'm supporting the Entomological Society when I'm selling insect stuff, and Mycological Society when I'm selling mushroom stuff. So at the end of the, like, usually December is a very good time to sell a lot of stuff because of Christmas and things like that. But yes, so yes, I just want to say thank you.